Professor Jack Suzinski is Allard Chair and Professor in Experimental Oncology at University of Alberta, Canada. He received his MSc with distinction in physics from the University of Poznan, Poland in 1980. He received his PhD in condensed matter physics from the University of Calgary in 1983. He did a postdoctoral fellowship at the University of Calgary Chemistry Department in 1983. He served as an assistant professor at the Department of Physics of the Memorial University of Newfoundland from 1983 to 88, and at the University of Alberta Physics Department from 1988 to 1990, an associate professor from 1990 to 93, and a full professor since 1993. He joined the Division of Experimental Oncology within the Cross Cancer Institute as the Allard Chair in 2005. He is on the editorial board of the Journal of Biological Physics, Journal of Biophysics and Structural Biology, Quantum Biosystems, Research Letters in Physics, Water, a multidisciplinary research journal, and Interdisciplinary Sciences, Computational Life Sciences. He is an associate editor of the Frontiers collection, Springer Verlag, Heidelberg. The major thrust of his computational biophysics group is in silico drug design for cancer chemotherapy applications and in vitro testing. Dr. Suzinski's research interests are strongly linked to the protein tubulin and the microtubules assembled from it. These have been studied using methods ranging from simple stochastic models to detailed molecular dynamics computer simulations, as well as through laboratory manipulations of living cells. Due to its prominent role played in eukaryotic cell division, tubulin is an important target for anti-cancer cytotoxic treatments. Their ongoing research aims to identify variants of known compounds showing greater tubulin isotype-specific effects. These could potentially lead to more efficacious chemotherapy treatments with lower side effects. Other studies by his research group have examined microtubule electrical, structural, and mechanical properties, proteins that bind to microtubules, and the motor proteins in cells that travel along microtubules and actin filaments. All of us are eagerly waiting to hear Professor Suzinski's inaugural talk entitled Molecular Models of Information Processing, Memory Encoding, and the Action of Anesthetics at the Level of Individual Neurons with Connections to Neurological Disorders. Professor Suzinski, please.
I will not be talking about anything to do with oncology today, although this is what I spend most of my time on, but because of the connection to this center, I'll try to spend some time discussing the role of microtubules in information processing and mental processes. And towards the end of my talk, I will try to make a link to some neurological disorders which we are actually currently working on. The usual, first of all, I'd like to acknowledge my sponsors. There's a lot of them. The research costs money, so I know that India is putting a lot of money into research, which is great news. We try to do our best too. And various collaborators from both Canada, Israel, USA, and at the end of this list you see Deepak Chopra, who's actually a co-author on our latest paper, which will be submitted today. And this is my group currently, and just two books that I authored or co-authored that have deep connections to consciousness. The first one, Emerging Physics of Consciousness, an edited book with Elizabeth's contributions, and I think Vishal, you also contributed. So I will try to make a point today, looking at the human brain from a different perspective, more technological perspective, that this is still the world's fastest and most portable supercomputer, not just a computer, a supercomputer. And I'll try to make a point showing you that actually it beats any supercomputer that we know, and by many orders of magnitude, with various capabilities still available. So what is the human brain in terms of computational capability? Well, there's 10 to the 11 neurons in our brain, and roughly 10 to the 15 synapses. At the current paradigm, this is where most of the information processing is taking place, but we'll go deeper, and obviously I think some of you will agree with us that microtubules will play a role, so we will go inside the neurons. When you compare it to, for example, the best supercomputer, the Blue Gene, which performs in 10 to the 14 flops, you'll see towards the end of my talk that actually we can easily see the brain as outperforming the Blue Gene by many orders of magnitude. Also, in terms of the energy consumption, which is non-trivial, the human brain consumes on average about 25 watts, which is less than a typical bulb. And the blue gene requires 1.5 megawatts, which would probably power a large portion of the city. So the question is there anything special inside each neuron that warrants a deeper investigation of the human brain's capabilities? And my answer to this question is yes. And I will not even go into quantum computing, which obviously is our goal, long range. But even at the classical level, the human brain composed of neurons that exchange information is an incredibly powerful piece of information processing machinery. To see this, I think we also have to look at the hierarchy of spatial dimensions. So as a physicist, I will try to simplify things a bit, maybe too much, but I hope you'll forgive me. When you look at the simplest organism, it is composed of about 10 to the 20th atoms, and we are in the realm of thermodynamics. Classical thermodynamics is sufficient to understand perhaps simple bacteria. If you go into cells, individual cells, then you see much smaller numbers of atoms. Already here, we are looking at possibilities of classical concepts being maybe not sufficient. And cells are composed of smaller organelles and subsystems of about 10 to the 5th atoms. And further down, biomolecules with thousands of atoms and individual molecules, tens of atoms. So here at this level, we definitely enter into the range of quantum chemistry and possibly quantum physics. The energy scale can be correlated with the scale of chemical kinetics. So the top bar on the scale is the binding energies of various bonds, and at the bottom is the KD values, dissociation constant. And, well, covalent bonds are the strongest, and hydrogen bonds would be the weakest, of van der Waals interactions. One thing that I want to emphasize here is that almost all these processes that we consider part of the living systems are non-equilibrium processes, so they require constant energy consumption and production. So it's important to understand the GTP and ATP hydrolysis. And for microtubules, it was already mentioned today by Pushta Sani that microtubules are operating on the principle of GTP hydrolysis. Their molecular behavior is driven by that. It's actually a very small amount of energy, just about 1.5 kT, just above thermal noise, but small enough to be considered quantum. I want to draw your attention to the work of Günther Albrecht Buehler from Chicago. If you have time, Google cell intelligence, and this will pop up as the first hit. He spent about 25 years trying to understand individual cells as intelligent systems. So we commonly think about organisms as intelligent, where intelligence starts, but Günther Albrecht Buehler shows us that actually individual cells already have intelligence. They are involved in very complex processes of perception and reaction to stimuli that require intelligent processing. And one of the observations that Günther Albrecht Buehler made is that cells respond to particular frequencies of electromagnetic radiation in the near infrared range around, let's say, 900 nanometers. And he actually demonstrated this directly by fooling cells into believing that there is another cell in the neighborhood, and he used infrared light instead. Importantly, I want to finish this thought. Importantly, in perception, there are two key aspects. One is sending the signal, so emission of a signal, and receiving the signal. So the question is, what is in the cell? What parts of the cell are capable of doing this? And Günther Albrecht Buehler pointed out to mitochondria as emitting signals. mitochondria produce energy, and energy production is also associated with electromagnetic energy emission. And the deeper question is actually what is receiving these signals, and he suggested centrioles are. And centrioles, as some of you know who are versed in biology, are composed of bundles of microtubules, 9 triplets of microtubules surrounding a doublet. So once again, we see the possible role of microtubules in the most important processes at the core of living systems. I'll go fast because Pushpa gave us some very nice introduction to microtubules. My objective here is actually to go into what microtubules do in the cell, but we need to know what they're composed of. So it's also important to distinguish between microtubules playing different roles in dividing cells and non-dividing cells. And it's also part of my research is most of it is dividing cells towards cancer research and non-dividing neurons, so it's consciousness research. But in both of these systems, microtubules are at the core of the cell's behavior. So transport, and information processing, motility, movement, and cell division in dividing cells. Organization is quite different, as you can see, from radial in dividing cells to parallel bundles in neurons. And we've seen this before, so in dendrites, there is a distinction between axons and dendrites. In dendrites, actual microtubules are antiparallel oriented, so they have a polarity, both physical and biological polarity, which Dr. Sani mentioned, namely positive plus ends or positive ends grow faster than negative, but they also have electrostatic differences. So there's a physical difference between the plus end and the minus end. So the big challenge for physicists and scientists in general is how you integrate all of this organization into a hierarchy. The hierarchy consists of structures within the neuron, such as bundles of microtubules and dendrites, interconnected by microtubule-associated proteins, MAPs for short, and an ensemble of other proteins, such as motor proteins, kinesin and dynein that participate in transport, and many, many other. I think there's probably 200 proteins at least that interact with tubulin. I've seen a list of 200, maybe more. So how all of this works in synchrony with a very high level of fidelity and organization. And the challenge is to go deep down to the level of atoms, because even single atoms may make a difference. And currently, actually, we're in a very fortunate situation, the first time in the human history, that we can ask these questions and even try to answer. This is a cartoon of microtubules playing a passive role, namely providing roadways for transport of material, kinesin. In neurons, this material is, for example, required to make neurotransmitters, pieces of RNA, and other material used for signaling. And we heard about dynamic instability. This is actually a computer simulation of a dynamic instability of microtubules. So you can see, speed it up a bit, neurotic microtubules going crazy, but they, left to themselves and given enough energy, they will be performing these very dynamic processes of growth and shrinkage. This is actually a very short video produced at Berkeley by Eva Nogales and collaborators who illustrate here the lifetime of a microtubule, and in about a minute you'll see how it all comes together. This is a tubulin dimer, the building block of microtubules. It's a stable unit that is now integrated into a microtubule structure, which is a cylinder, and now this is the dynamic instability catastrophe, showing how microtubules disassemble, the protofilaments fray apart. This is a stable ring. A ring of tubulin will constitute the nucleation center. There's also gamma tubulin that can be used artificially to nucleate microtubules under various conditions. That's the surface of a dimer, tubulin dimer, and hydrolysis that was mentioned before actually has a role in changing the conformation. So the dimer changes shape as a result of GTP losing one phosphate and losing energy, and that makes microtubule unstable. It's a little bit like building a very faulty tower. Simply the bricks don't fit and they will collapse. House of cards, in other words. And that shows you also beautiful crystallographic structures. Microtubules are not just cylinders, actually. They have a perfect lattice, two lattices, in fact, so-called A lattice, which is perfect, and B lattice, which is not. Here you also see pores between tubulin. There's openings or holes, and these holes play a non-trivial role. If I have time, I'll show you what we believe is happening in these holes. Some experiments were done in Spain that suggested that the holes actually are very important in transport. This is, again, folding back to microtubule into a cylinder from millions of tubulin dimers in solution. One of the processes shown here is the folding of a sheet into a cylinder. This is probably the dominant way of forming microtubules. And this is microtubules in an aster formation. And here's, again, the folding of the sheet, zipping up. So there is a very interesting set of dynamical processes. A lot of people are, but I think not that many people are studying them from the point of view of chemical kinetics. So your goal is very noble. I think it'll be a very interesting piece of research. This is the group that created this, thank you, this video. So, and then we go, however, now we go back to the presentation. It shows you the dynamics of these processes and the incredible synchronies taking place. But we can go deeper, actually. We can go into the level of single atoms. We now know, since 1997, what the structure of dimer is, tubulin dimer, in terms of atomic positions. And we can simulate the behavior of these atomic coordinates to great level of accuracy. And so back to the idea of zooming in or hierarchies. Here the hierarchy is the dimer forming a lattice, and this is a micrograph of an actual microtubule, and again fluorescently labeled microtubules in a cell. And you can also see different dimensions, 4 nanometers, 25, 10 microns. This is so this one, if you can see it, actually, I have to click again. It shows individual atoms vibrating due to thermal noise, because all of it is actually in solution in the body, in the human body or other cells, at physiological temperature, 36 degrees Celsius or so, right? So this temperature causes all these atoms to vibrate. So it is really not a static picture, but it's a dynamic. And again, so this is the crystallographic structure with all the alpha helices and beta sheets. And we now have several files, coordinates of atomic structures from different experiments in France and the US. And these are listed here. There's more now. They were obtained under different experimental conditions, and it shows how they fit into the structure of a microtubule. So that's from X-ray and electron crystallography. And for me, it's important that all these images were obtained from experiments involving some important molecules in cancer chemotherapy. Molecules such as paclitaxel, which is used in curing breast cancer, ovarian cancer, lung cancer. Also zinc alkaloids used in leukemia treatment, lymphomas. So that is, has an additional value that we can actually study the interaction of known chemotherapeutic drugs with tubulin and improve on this. But again, that's all I have to say today because it's not our objective. So what I'll show you is a lot of pictures from computer simulations based on the input from crystallography. So biochemistry can give us a lot of information that we can then transform. That shows a protofilament formed from tubulin dimers. And you might be wondering what these protrusions, what these tails or horns are doing there. These are actually quite flexible structures called C-termini. C-termini, proteins have N-termini and C-termini. Tubulin has C-termini 2, 1 per monomer, and they actually effectively extend outside, making the microtubule much bigger effectively than it is, because C-termini are an additional area that interacts with other proteins. And this is how you make a lattice, by putting protofilaments together, and you can see the flexibility of protofilaments. These bonds are stronger, so you can recall now the fraying of the microtubule protofilaments on catastrophe and see why it is so, because these bonds are stronger than the other ones. Sorry, I meant weaker, so the protofilaments don't bind so strongly as dimers within the protofilament. And these are the two lattices that are obtained by putting protofilaments together, the so-called A lattice and the B lattice, which has a seam. We still don't really know if there is any biological function to the seam or the difference between the lattices. A lattice is more common than B lattice. No, sorry, the B lattice is more common than the A lattice, but we still don't know what function this might play. And again, physics may come in to help us, because this actually is a result of calculation, free energy calculation of the entire microtubule lattice, showing you that there are two energy minima, each of which corresponds to a different structure. So based on physical calculations, you can predict that only two lattices are stable with 13 protofilaments. And again, you can put it back together and build it more or less like Lego using computer simulations in 3D. And then this is the entire microtubule reconstructed using computer models. It has millions of atoms in it. It's actually at the level of individual atoms. You don't see them, of course, but they are there. And C-termini decorating the surface. And here it's a cross-section of a microtubular ring with the C-termini sticking out. I want to pay special attention to it, because in our opinion, C-termini are very, very important. For one, it's now experimentally demonstrated that if you cleave them, if you remove C-termini biochemically, no microtubule will ever form. So paradoxically, they don't make any contact between dimers, but they are critical in making microtubules. Probably they keep the structure together. And this is three different views of a tubulin dimer, again reconstructed in the computer with various important motifs. I want to point out to the GTP molecules that one of them is causing instability. The M loop, which is actually a result of instability, switches two positions. It's almost like a switch of the light on the wall. You switch it from here to there, and the molecule is undergoing a conformational change, and microtubule collapses. So we know quite a bit about how things work there. And finally, in connection to cancer research, we know exactly where this paclitaxel molecule or taxol sits, and therefore we are now capable of designing better drug molecules if we want to. This will come back later on, but I want to tell you that we also know where other molecules bind to tubulin, and these are the locations. Vinblastine, vincristine are binding to the top of the beta tubulin, paclitaxel on the inside of microtubule surfaces, colchicine at the interface. And interestingly also, all of these molecules actually come from natural sources, from plants. And it's one of the mysteries that plants, maybe to us, and I think different civilizations have different points of view. In the West, it seems a mystery that plants have made all these wonderful drugs without our intervention, right? I think in India, maybe in China, people take it as something normal. not a big deal. So all of it actually has also to do with electricity. Believe it or not, what I showed you so far were cartoons, but there is also electric charges. This protein has a lot of electric charges, most of them actually on the C-termini, and these charges make particular structure possible. This cartoon shows you so-called equipotential fields of equal potential around a tubulin dimer. And that shows you how this explains, for example, the ring formation. Gamma tubulin is shown here, making a ring. Blue means positive charge, red negative, and you can see how complementary the charges are in this arrangement, and therefore energy is minimized. So this is a natural state of gamma tubulin in a ring to minimize free energy. This is the entire microtubule. Maybe it's not so obvious here, but it is also an energy minimum structure. This is, again, a microtubule painted with charge in the same, with the same convention. And you can see two ends of the microtubule are different electrostatically as well as dynamically or biochemically. And one more thing that is kind of intriguing and prompted us to look deeper is that across the surface of microtubule, which is about 5 nanometers thick, there's 100 millivolt potential difference, with the outside being more negative than the inside. And this is really almost identical to membranes and ion channels across membranes. And microtubules have also their own ion channels, passive, which we call nanopores. You see these holes? Well, these holes are about 1 nanometer in length and 5 nanometers in depth, and about 100 millivolts across in terms of voltage difference. So ions, depending on their charge, will have preferential flow either inside the microtubule or outside the microtubule. And we've done simulations on this. Okay, so before I show you the simulations on ion flows, I also want to spend a few minutes on C-termini, because C-termini, in our opinion, are absolutely critical to microtubule functioning as information processing devices. Why is that? These are the most diverse structures in tubulin. Today we heard a talk about evolution, how evolution across billions of years was instrumental in bringing intelligence to our planet, and how evolution maintains useful things and discards things which are not useful. Microtubules are almost as long as life on Earth, about 2 billion years. And actually, tubulin is highly conserved. So from the simplest organism to our human brain, which is the most complex structure known to us, at least, tubulin has been almost always the same. This is a mystery in its own right. Therefore, it must be very useful, must be very good, intelligent, etc. But one thing that has changed, when you look at the evolution of tubulin from, let's say, the simplest organism, Paramecium, that has tubulin, to the human brain, one thing has changed, and that's C-termini. Every organism has a different C-terminus, and even in the human body, you have 20 different types of tubulin, each of which can be recognized by the sequence of amino acids in the C-terminus. So it's an address label. You can look it up and say, oh yeah, that's from a chick. No, that's from a pig. No, that's from a human. Just by looking at the C-termini. It's only about between 10 and 18 amino acids depending on the species, and yet it contains crucial vital information. You can make antibodies for tubulin based on the C-termini. And secondly, they are very negatively charged. So most of the electrostatics is in the C-termini. And finally, they interact with almost all the proteins. So the proteins that interact with tubulin always interact with C-termini. Kinesin binds to C-termini, MAP, tau, MAP2, all these microtubule-associated proteins which make dendritic microtubules in neurons or maps in axons, they also bind to C-termini. So there must be a recognition process at play. Now, we have studied the C-termini also from the point of view of their dynamics. And it's very interesting because of the negative charge on C-termini and the negative charge on the surface, their preferred orientation is like this, with basically standing up, to C-termini up. However, there is a positively charged patch on the surface. I don't know if you can see it. Maybe there'll be a better picture later. But around here, there are some positive charges. So C-termini can bend over and bind. So in other words, this is a two-state system. Actually, it's a four-state system because you have alpha and beta. So physicists in the audience will recognize this as somewhat reminiscent of the Ising model, right? Spin up, spin down. Except here we have a field. There's a preference for the spin up compared to spin down. Spin down has a probability of 15%, we calculated, and spin up has a probability of 85%. So it's almost like a biasing magnetic field. And this is supposed to be a simulation showing how, we'll see if it works. Our simulation, how C termini actually dynamically can switch from one state to the other. Now, why am I talking about this? We can probably go to the video. It's called, no. Why are we talking about this? Because in my opinion, This will show you in a few moments how you can unbind a C terminus from the surface. At the bottom, this is at the top is looked from above. Oh, now it's freed from the surface. And there's another video that shows you the opposite, that can after a while bind back to the surface. So first of all, there is the influence of the environment on this dynamics. pH, ion concentrations, etc., temperature will affect this process. So the microtubule will know what sort of environment it's living in. Secondly, this will be responsive to electromagnetic fields. Electromagnetic fields will be able to deliver energy to change this. I think you can store a lot of information in it. Of course, energy might be needed for it, but nonetheless, it's one of the degrees of freedom for storage of information. And it's a binary state, up and down. So you can think about it as the beginning of a computer, or a bit. I mentioned the isotypes. Isotypes are different tubulins encoded by different genes. And in the human body, we have at least 20 different isotypes. So it's interesting how they are distributed. And they play a role because different cells have different distribution of tubulins, and you can tell them apart. So for example, beta-3 tubulin is very dominant in the brain, and we don't know why it is so. And it's also found abundant, or to some degree abundant, in all cancer cells. So the question is why? And as we all suspect, life makes choices for a reason. It's probably not a random choice. If you have a repertoire of 20 tubulins, why do you choose this one? So I showed you this primitive dynamics of C termini. We've actually applied it to computer simulations of ion flows and simulated the whole microtubule as if it was an piece of electronics. Electronic chip. And further, before I show you the results of the electronics simulations, we also found that every single C termini sequence, so every isoform of tubulin has different sequence of C termini, and each of them has different frequency and different mode of vibration. And these, I'll show you a few of them. So every microtubule, depending on where it comes from, a human microtubule or again, a bovine microtubule, will have a different dance. One dances the tango, the other dances the waltz. And this is only a metaphor, but I think it's actually will be responding to different frequencies and therefore resonant with the environment to some degree. Basically, every C terminus has a different mode of vibration, right? So let's summarize it. Statically, it's up or down. They can switch between the states, but they can also, when they are in one of the states up, they can also dance around. Okay? And if you have a microtubule in a cell, for example, a liver cell will have microtubules composed of several types of isoforms. Now you can think about a dancing floor with people simultaneously dancing the tango and the foxtrot. Okay? So you can mix and match and start building complexity here. So we went a bit further and did some experiments with my collaborator Horatio Cantiello at Harvard and Avner Priel in Israel. And we did experiments on microtubules and their conductivity. So that was kind of out of the blue. I think most people would not think about microtubules as conductive elements, but actually they are. And here is a picture of a microtubule held by 2 micropipettes by Horatio. And I think he's probably one of the very few people in the world who can actually grab microtubules with both pipettes. This is about, I don't know, 20 microns or so. And these micropipettes were wired, so you could send an electric signal, a pulse, a voltage pulse, and this pulse traveled, and then you can measure the pulse at the other end. And these are examples of traces of currents as a function of time. And what's interesting is the microtubules actually behave like transistors. They amplify the current. So you inject a voltage difference of a particular value, 100 millivolts, and then it actually grows as it propagates from one end to the other. So actin filaments, I have no time for this, but Horatio can tell that similar experiments on actin filaments, they don't amplify, but they pass these signals faithfully. In other words, you can start building electronics from actin filaments and microtubules with rectifiers and transistors. Amplification and rectification of these currents is possible. So suddenly you have a system that allows you to work as almost a microelectronic chip inside the cell. So we've built computer models how this works with ion flows. We believe these are actual ionic clouds congregating around microtubules. Because of the negative charge on the surface, positive ions, such as sodium and potassium, will be attracted to it in a cloud, but they cannot land on it, all at least. And they will then, on stimulation, propagate. And on top of that, remember the holes. These holes have potential differences as well, so ions can flow around into the hole and inside the lumen. So it's a very interesting and non-trivial piece of electronics. And this is an equivalent electrical circuit that we built that allows you also for flow of ions across the pores inside the lumen and from end to end. And we created these microtubules IV characteristics, which are typical of transistors with uneven portions, whether you send the current one way or the other, which is actually what experiments demonstrate. And so I'm going to switch gears a little bit. This gave you a little bit of taste of where we're going with this. There are several issues, one of which is how the cell uses these structures for signal propagation. Another question is, can you use it for storage of memory, or some information can be stored? And this actually goes back a long way to Stuart Hameroff, who was mentioned a couple times today. Stuart is a good friend of mine and a visionary, a unique person. You will see him in December. And he's just amazing. He came up with this idea that microtubules can actually store information about 20 years ago without knowing anything about the structure of tubulin at that time. Now we know it, and I think we can say with some conviction that what he predicted is coming to pass. So this is my very quick summary of where I see the storage of microtubules and how much information you can store in the brain, how you can compare it to the supercomputers of today. So if you count the states on tubulin, you have 4 states per dimer in terms of C-termini, up and down at least. You have also electrons which can hop between 2 positions, and then you have 2 conformational states, GTP-based, GTP hydrolysis. So you have 4 times 4 times 2. It's 32 states per dimer, at least, conservatively speaking, which is, if you calculate the number of microtubules, which was listed in one of the talks today, it's about 100 kilobytes per microtubule, or 1 gigabyte per neuron. I have a memory stick in my bag, which has 500 gigabytes, but it's that big. A microtubule has 1 gigabyte, at least, and it's 25 nanometers across and 10 micron long, okay? Now, if you do the algebra, it's about with 100 billion neurons, it's 10 to the 20th bits per brain. And if these states, as we know, these C-termini states and the electron hopping states can change on the time scale of 1 nanosecond. That is 10 to the 28th flops per brain. Our brain, based on this picture, is capable of executing 10 to the 28th flops. The blue gene, IBM's pride and joy, has 10 to the 14th flops, right? Now you see the difference? 14 orders of magnitude. It does 14 times, 14 orders of magnitude. The question is there a way of storing and recalling information? So there are different types of information in biology, and there's genetic information. Everybody knows about DNA, and there is information in the brain that we are talking about now. We still don't know exactly where it resides, but in principle, it can last 100 years for as long as we live. We have immunity. Immune system has a memory of about 10 years, and we have reflexes, which are sort of immediate responses. So it's about 1 second or so. The persistence of biological memory, therefore, can vary from seconds to billions of years if you take into account evolution as some sort of memory of the entire planet. Now, this is kind of interesting. When you read the literature on human memory of our brains, you'll see huge differences in opinion about this. Interestingly, the older you go, the more optimistic the people were, and more recent accounts are completely pessimistic. And I believe von Neumann, actually. Von Neumann estimated that the human brain can store up to 3 times 10 to the 20th bits. Remember my estimate? It was 10 to the 20th, very close. So I agree with John von Neumann. He calculated how much we can store in terms of all the information that we gather throughout our lifetime, and that was the number. So you can, if you use microtubules in the brain as storage devices, you can store this in microtubules. We don't know exactly where. I'm speculating that these 4 different modes of information processing exist. Now, when you look at more recent estimates, Landauer was completely, I don't know, pessimistic about the human race. 10 to the 9th bits per human brain is ridiculous. So according to Landauer, we are even not as good as a memory stick for your computer. I mean, come on. And I've heard some talks about spirituality and religion. Some of you may have heard and read about near-death experience, right? People that thought they were dying, they came back to life because of various, modern techniques or otherwise. They were resuscitated, and they tell stories about this life review. The process in a very short time, they go through the entire life in great detail. How many of you read about this? Yeah, you know about this, right? So now we have documented cases of thousands and thousands of people going through something very similar. And it's almost like unwinding of a tape. It's sequential. And I think if you think about mapping information in microtubules, you could also see this as a possibility of unwinding the information stored in microtubules. That may be simply releasing all of this, perhaps in the form of electromagnetic emission, rather than biochemical or physical, encoding. Now it's decoding, perhaps through a flash of electromagnetic waves. This is kind of a list of different sources of information, and our human brain, if you believe me, has almost infinite potential. We could probably remember, if we wanted to, we could remember all that has ever been written or stored in our brain. And there are some examples of idiot savants, people who have these capabilities of instant recall. They can memorize almost everything instantaneously and remember for the whole of their lives. Or hypnotic situation. Under hypnosis, you can recall a lot of things that you don't remember anymore. I used to speak fluent Russian. I don't speak it anymore, but I'm sure under hypnosis I could speak fluently again. So here I propose that tubulin is a memory chip. It is a storage device with several modes of storage could be long-term and short-term. Some of it may be easier to retrieve, some of it may be harder to retrieve. Maybe C termini are easy to retrieve because they have contact with the environment very easily. Now, we have also proposed recently with Stuart Hameroff and Travis Craddock a very specific mechanism how this can link to the known experiments on animals with memory recall. And this was published in 2008, where scientists were able to erase memories in mice under controlled conditions. And these experiments indicated that there is a particular molecule or molecular complex, calmodulin kinase complex 2, that is directly involved in memory encoding and decoding. And this complex is formed in dendrites in the synaptic connections, and it actually requires calcium to be activated. And it's used to phosphorylate tubulin. So calmodulin kinase 2, and phosphorylation, as you know, in biology, in cell biology, is a very common process for signaling or tagging molecules for a function. So this one, CAMK2, phosphorylates tubulin. We claim that we know how it does it. And I'll show you some pictures which illustrate it. Pictures, again, are generated by computer simulations. This is how this complex is made. It first has to be made by calcium molecules and calmodulin. An inactive complex doesn't have these. Active has calcium and calmodulin. And so the question was, and this is the title of the paper which we published and with Travis and Stuart in Journal of Integrative Neuroscience last year, that there are phosphorylation sites on tubulin in microtubules, and these phosphorylation sites actually correspond very closely to the calmodulin kinase 2 complex geometry. So these are the areas where phosphorylation occurs. This is the complex which we build in the computer. It actually looks like a hexagonal robot. What's the name of this toy? Transformer? Like a transformer, kind of. Stuart Hameroff called it a nano poodle. You know, French dog poodle. It looks a bit like a poodle, and it's a nano size. But it fits very well into the microtubule structure. Now you see, in gray you have a patch of tubulin on a microtubule, and the color pictures are of the calmodulin kinase complex. And remember two lattices, A and B? So you can twist the complex to fit into either of these lattices. But of course, it's easier to in the original form, it's easier to fit into this hexagonal lattice, but both possibilities are available. And now you see the nanopoodle walking on a microtubule, and as it walks, it actually phosphorylates sites, so it leaves tags, and that's a permanent memory until it's erased, of course. And we've created a logical table of phosphorylation sites on microtubules. So this is mechanistic. Red would be phosphorylated sites, and so because we have no phosphorylation, a single phosphorylation, or a double phosphorylation down on alpha or beta, we called it trites instead of bytes, right? Because we have 3 states, so now we have instead of 2 states, so you can build a whole logic around it. And if you do the calculation again, that alone would have 10 to the 19th bits per brain. We claim this actually has to do with short-term memory, perhaps, because it's something which is erasable very easily. Just like in these experiments with mice, it was reversible on stimulation. I'm almost done. I want to finish with something that is of great interest. Alzheimer's disease. In the West is a very growing problem because of the aging population. There's an incidence of Alzheimer's now, I don't know, 20% of people in the 80s and so on will have it. We still don't know what causes Alzheimer's. We believe that it has to do with zinc and competition for zinc binding between tubulin and beta amyloids. Beta amyloids are proteins in the extracellular matrix of neurons, outside of neurons, and zinc promotes their aggregation. But microtubules require zinc for building new microtubules. And you saw the microtubules being dynamic. They are not static. They constantly grow and shrink. And even in the brain, in your neurons, every 6 hours, new microtubules are growing and the old ones are shrinking. So now suddenly, if you don't have zinc to participate in this process of regrowth of microtubules, your neurons will be slowly dying because they will be losing microtubules and the structure will disintegrate. So we created a model. With Rudy Tanzi from Harvard, who is a renowned neuroscientist, which basically demonstrates that the stealing of zinc by beta amyloids has a double effect. First of all, the growth of plaques in the brain, and secondly, the disappearance of microtubules and consequently the death of neurons. So now, if that's correct, we have a clear target for drug design. How do you design drugs now? The drugs would have to basically cover the sites where zinc binds on beta amyloids and release the zinc to go back into neurons and regrow microtubules. So I'll just say 2 words about it, and I'll close my talk. So one more question that was asked is anesthesia, and as you may know, Stuart Hammer is an anesthesiologist. So he knows anesthesia from day-to-day activities in his professional life. Anesthesia, of course, one of the aspects of anesthesia is instantaneous memory loss. And the question was, do anesthetics bind to tubulin? And so this one segment that I didn't have time to talk about, actually we did calculations and showed where anesthetics bind on tubulin. And in one sentence, anesthetics bind roughly in 3 locations where I showed you where different drugs, zinc alkaloids, paclitaxel, and colchicine bind, and also to C-termini. So they may block interaction with other proteins, and they may also block C-termini dynamics. So that's basically all I had to say today, and I thank you very much for your attention.